Hi everyone, welcome back to my A-level biology lecture series. In this lesson, we will explore the tryptophan operon, which is a repressible operon. Let's get started. The tryptophan operon in E. coli is an example of a repressible operon that regulates the biosynthesis of tryptophan. And tryptophan is an essential amino acid for bacteria to grow. So the component of tryptophan operons are promoter, operator, and structural gene, which are TRPE, TRPD, TRPC, TRPB, and TRPA. So in this diagram, you can see that the tryptophan operon consists of the promoter. Here we have a slight mistake. The green color is the promoter and the yellow color is operator, and then followed by 5 structural genes. So the gene TRPE and TRPD encodes the 2 subunits of enzyme 1, means that enzyme 1 is a quaternary protein, while TRPB and TRPA gene encodes these 2 subunits of enzyme number 3, and TRPC code for enzyme number 2. So enzyme 1, 2, 3 involved in the biosynthesis of tryptophan, from the precursor to the intermediate compound, and finally we have tryptophan in the bacterial cells. So under normal condition, when tryptophan concentration is low, this operon is always switched on. However, when tryptophan concentration is high, tryptophan now acts as the co-repressor, or the repressor that actually stop or inactivates the tryptophan operons. So therefore, tryptophan operon is a repressible operon. So yep, enzyme 1, 2, and 3 completes the pathway to produce tryptophan. There is a regulatory gene which is located away from the tryptophan operon, and it has its own promoter. If you compare back with the lag operons, the lag operons regulatory gene is nearer to the lag operons. However, in the tryptophan operons, the regulatory gene is located away from the tryptophan operons. So with the promoter, this regulatory gene is transcribed into mRNA, and after that translated into a repressor protein. However, this repressor protein is inactive by default, so therefore this repressor protein cannot bind to the operator. So when tryptophan concentration are high, tryptophan now acts as a co-repressor by binding to the repressor allosteric site. So this changes the conformations of the repressor protein and activate this repressor protein. Activated repressor protein enable this repressor protein to bind to the operator and therefore block the transcriptions of the structural genes. And this is correct, and it makes sense because if you look at this, the repressor protein is inactive by default, so that these 5 structural genes can be transcribed and form the enzyme that needed for the biosynthesis of tryptophan. When the concentration of tryptophan is high, and tryptophan now become the co-repressor, bind to the allosteric sites of this repressor protein and activate the repressor protein. And when this active repressor protein binds to the operator, RNA polymerase now no longer can bind to the promoter, therefore stop the synthesis of the enzyme that needed for the tryptophan synthesis. So in bacteria living within a host, when the environment lacks tryptophan, the tryptophan operon is switched on. So the cells activates the metabolic pathway to synthesize tryptophan from the precursor molecule. Precursor molecule means the starting molecule. When the host diet provides abundant tryptophan, therefore the tryptophan operon now is switched off. And the bacterial cells stop the production of tryptophan, conserving energy and resources. So this is an example on how bacteria adjust their metabolism in response to the change in the environmental nutrient availability. And with that, we have come to the end of this lesson. Hope you found it helpful and easy to follow. If you have any question, feel free to drop them in the comment section. See you in the next lesson. Have a great day.